In eukaryotes, a cell cycle is a series of events that occur in tandem with cell division and proliferation. Typically, this is the representation of human cell cycle, which consists of 4 phases: G1, S phase, G2, and mitotic phase. DNA replication occurs in S phase. DNA replication is the process to synthesize a new DNA. DNA replication is strictly regulated to ensure the process happens only once per cycle. What controls DNA replication? CRL4-CDT2 ubiquitin ligase is the key regulator of DNA replication. One of the vital proteins is CDT1, which is a licensing factor that functions to limit DNA from replicating more than once per cycle. A study confirmed that a fragment of C-terminal CDT2 460 to 580 has DNA binding activity. Thus, this study aims to transform the plasmid harboring C-terminal of CDT2 460 to 580 DNA binding protein into the competent cell for further analysis. This study also aims to determine the expressed plasmid for DNA sequencing. These are the list of materials and apparatus used in this experiment. This project starts with preparing LB agar and LB broth as media. LB broth and LB agar are commonly used in microbiology to grow and study bacteria like E. coli. Then, the preparation of competent cell was done through calcium chloride method. The calcium chloride method allows the plasmid to be introduced. Next, transformation takes place when competent cell uptake the DNA from its environment. Through heat shock method, the sudden rise in temperature will produce pores in the plasma membrane and will allow the uptake of DNA. Next, the selection of positive transformants is possible due to the antibiotic-resistant genes carried on plasmid. To confirm positive transformant after transformation, scale-up culture was done. Plasmid extraction was done using the prime-wave plasmid DNA extraction kit. The plasmid extracted is analyzed through the gel electrophoresis. Before preparing the competent cells, gel electrophoresis was run to ensure the presence of the DNA plasmid in the sample. The DNA plasmid on the gel were compared to 1 kb DNA marker. Hence, the band on the gel under UV shows the presence of the plasmid. However, the DNA ladder show curve and slight separation on the bands, which cause it harder to interpret the accurate base pair. The smear might be caused by overloaded amount of DNA ladder or damage to the wells when the sample was loaded. If the sample was loaded in high volume, it may cause overflowing from wells. Meanwhile, the poor separation of the DNA ladder may be caused by a slow run. Thus, to improve the visualization of the gel, load less amount of DNA ladder to avoid overflowing. Besides, load DNA ladder after all the samples have been loaded to get good visualization. For the first trial, the cultures observed were less cloudy even after 2 hours of incubation with 1200 RPM. Thus, the incubation was continued until 4 hours with increased RPM, which is 2500 RPM for the second trial, to allow the bacteria grow rapidly with the increase of oxygen concentration. The second trial shows the cloudy appearance of bacteria cultures after incubation for 4 hours. Transformation of plasmid harboring CDT2 480 to 560 into competent cells was done for 6 times due to no growth on LB ampicillin plates. No colonies on the plates may caused by several errors during preparation of competent cells and transformation. It may be due to low efficiency of competent cells. The laboratory only provide negative 20 degrees Celsius freezer, which caused the stored competent cells decrease in its efficiency over time. Thus, the competent cell should be stored in the negative 80 degrees Celsius freezer to maintain its efficiency. Furthermore, temperature is a crucial factor to consider when using heat shock method during transformation. Incorrect steps will cause the cells not be adequately heat shocked. In addition, using a dry block heater may lead to an inconsistent temperature, which causes the temperature to be either more or less than 42 degrees Celsius. Hence, it is recommended to use a thermometer to check the temperature of the dry block heater or replace it with a water bath. For the 7th trial, the plate showed 2 visible colonies. After the possible causes have been troubleshot, a positive transformer was successfully transformed. The visible colonies on the LB ampicillin plate indicates bacteria growth with plasmid containing ampicillin-resistant gene. The gene allows the bacteria to grow and survive even with the presence of antibiotics. To confirm the colony is a positive transformer after the transformation, a single colony was inoculated and regrew in the LB ampicillin broth. The growth of the positive transformer after retreat on the LB ampicillin plate and incubated overnight was shown. To verify the bacterial growth is E. coli, gram staining was done. Gram staining enables the determination of bacteria through their morphology and arrangement. The result under 100-time magnification confirmed the bacteria on the LB ampicillin plate is E. coli. This is shown by the appearance of pink indicating gram-negative bacteria. After repeating gel electrophoresis twice, the results are still not showing any bands. This is made due to insufficient bacterial cells when the cultures were incubated. The cultures are recommended to grow at 37 degrees Celsius in a shaking incubator, but do not incubate more than 18 hours. Incubation after 18 hours may cause the cells to leave log phase and enter stationary phase. Afterwards, the bacteria cultures may lose the plasmid DNA during plasmid extraction. Unfortunately, the process cannot be repeated due to time constraints. Hence, further analysis of the plasmid cannot be done. In conclusion, the transformation of plasmid harboring C-terminal of CDT2 4602580 into the expression host BL21DE3 was successfully achieved. However, the transformed plasmid were unable to extract for DNA sequencing due to time constraint. In order to obtain positive results, these are the recommendations suggested.